My name is FGF1 and welcome to this episode of Draw My Life. So where do I start? I'm produced as a direct product of the FGF1 gene locus. In humans, that's located at chromosome position 5q31.3. No one has ever found me in unicellular organisms. I was initially discovered in brain pituitary extracts in a US laboratory in 1973, though I'm widely understood today to be expressed in cells and tissues from all three germ layers. You can see me helping out with cell proliferation, cell differentiation, cell migration, and even cell survival. I was named a year after my discovery by a man called Dennis Gospodarowicz, though when he first saw me I was isolated at an acidic pH, so he called me something funny. You're an acidic fibroblast growth factor, FGF, he would say, not knowing I had loads of other basic younger brothers and sisters, FGFs, who had not yet been found. I have 21 human siblings to be precise, FGF2 through FGF23, and FGF15 only exists in mice, but FGF19 carries out their endocrine functions instead. We all enjoy broad mitogenic and cell survival activities. There are a couple of us who can't stimulate fibroblast proliferation. They're my step siblings. We're only related because we have similar protein structures, roughly 30 to 50% amino acid sequence homology. I'm talking to you, FGF20, and subfamily FGF8. Me and my FGF siblings can be divided into 7 different subfamilies, each with distinct phylogenetic properties, sequence similarities, and functional properties. There's the FGF1 subfamily, that's me and my sister FGF2. We carry out local paracrine functions with the other subfamily groups, FGF4, 7, 8, and 9. Subfamily FGF11 and FGF19 are known as the odd ones out. FGF11 carries out intracrine roles, helping with intracellular signaling and the opening and closing of voltage-gated sodium channels, while FGF19 carries out endocrine metabolic functions. Weird. Structurally, I like to think I'm interesting. I am a single non-glycosylated protein chain weighing between 17 and 18 kDa with a total of 155 amino acids. They say I'm in great shape, not to mention my 12 anti-parallel beta sheets. These sheets make me a friendly face and they allow me to interact with my receptor friends really easily. There's plasticity in the interactions I have with receptors because I am generally very easy to get along with. Sometimes, I'm even referred to as promiscuous. Generally, I'm the accepted universal FGFR ligand, which speaks for itself really. Unlike my sister FGF2, who is released as a monomer, I'm released in an ATP-dependent process as a disulfide-linked dimer. Together, we love helping out in development. One of our roles as intracellular FGFs is to protect the cell against stress conditions by providing an additional signal for cell survival independently of receptor-activated signaling cascades. I don't want to brag, but we do a lot of the so-called heavy lifting. When something gets in the way of our job, it can result in a number of harmful cardiovascular, skeletal, and even neuronal defects. This makes us sad. It's not what we like to see. My other siblings are useful too. For example, my brother FGF7 helps with both the growth of hair follicles and the ureteric bud. One of my other sisters, FGF9, who's a bit of a control freak, helps with the development of the lung mesenchyme, heart, GI tract, skeleton, and testes. I don't get a lot done without the help of my receptor friends, the FGFRs as they're known. These guys are cell tyrosine kinase receptors, and together we comprise an important cell signaling system. I have 4 best friends, FGFR 1, 2, 3, and 4, and they all have 3 extracellular immunoglobulin domains, D1, 2, and 3, and a single transmembrane domain and a cytoplasmic tyrosine kinase domain. Combined, they can code up to 48 isoforms. Me and the other FGFs work together with our receptor friends, binding and dimerizing in one of 2 ways. We paracrine FGFs use heparan sulfate cofactors as binding partners, which stabilize the receptor ligand structure, enhancing our resistance to protein breakdown. Alternatively, my endocrine FGF siblings utilize clotho co-receptors in the receptor binding. Gain-of-function FGFR mutations have also been identified in a variety of cancers, including those of the prostate, breast, myeloproliferative syndromes, lymphomas, as well as other malignant diseases. I've got big plans for the future, and there has been a lot of research done to figure out us FGFs can be of clinical use. One Chinese study revealed that my close sister FGF2 can be used to accelerate healing and improve the scar quality of burns and the range of surgical wounds. I've also heard that the use of FGF methods have shown to be an effective way of treating chemotherapy-induced mucositis, an inflammation of the mucosal layers of the mouth. This side effect, when severe, can often interrupt chemotherapy treatment, so this application is well useful. Research is also currently underway investigating the use of drugs that inhibit the FGFR signaling pathways as potential cancer treatments. For example, the growth of squamous cell carcinoma cell lines can be inhibited with FGF2 ligand traps. Anyway, the future looks bright. That's all for me. Thanks for watching!